Thank you for giving this opportunity to talk about my research, which is focused on um, malaria and pregnancy. So we've already been introduced a bit of the Plasmodium parasites, and one of the species that is known to infect pregnant women is Plasmodium falciparum. An infection with this species of Plasmodium can lead to poor health outcomes in both the mother and also the accumulation of infected red blood cells in the placenta. Now, these infected red blood cells could they accumulate whether in the intervillous space here or also bind to these villi structures. Now, if you look closely at the interaction of the infected red blood cells and the host cells, we see that these infected red blood cells, they bind to receptors on the host cell. Now, one of the key receptors that has been well characterized is known as conjunctive sulfate A, or simply known as CSA. Now, the infected one protein that is a key mediator of this interaction is simply known as VAR2CSA. So this is a parasite-derived protein, is expressed on the surface of the infected red blood cells. So here is shown as these orange spikes. So what we know that antibodies that target this parasite protein known as VAR2CSA, they could provide protective function. And one way is by preventing the binding of the infected red blood cells to the host cells. Now, two key information I want to talk about that this parasite-derived protein VAR2CSA is only expressed in parasites that infect pregnant women. And similarly, antibodies that targets this protein can only be acquired during infection in pregnancy. Now, most of these studies, they've been conducted in sub-Saharan Africa, where the predominant Plasmodium species is Plasmodium falciparum. Now, what was surprising was that during one of our studies in Colombia, we observed VAR2CSA antibodies in non-pregnant populations. Now, when I mean non-pregnant populations, I mean in men, in children, and female below the age of 12. Now, this was surprising because it's well established that such antibodies can only be acquired during infection in pregnancy. Now, we know that in Colombia, you have the existence of multiple species of Plasmodium, but predominantly Plasmodium vivax and Plasmodium falciparum. Now, another intriguing observation we had was that antibody that was specific to a protein present in Plasmodium vivax, known as Duffy binding protein, antibody specific to this protein recognize VAR2CSA. So this indicates that there's a kind of a cross-reactivity going on between these two antigens. So this Duffy binding protein, the parasite uses it as an evasion ligand in its own cell. Now it contains some structural features that are also present in VAR2CSA. So we postulated that that exposure to this Duffy binding protein can elicit antibodies that also recognize VAR2CSA. So one of our key research questions is identifying these regions in VAR2CSA that are recognized by cross-reactive antibodies. And one approach we used is known as peptide array. So in peptide array, what you do, you generate overlapping fragments, and these fragments are printed on a chip, and we scan with a monoclonal antibody. Now this monoclonal antibody is quite special, so we acquired it through collaboration with one of our colleagues in the US. This monoclonal antibody was generated against this vivax protein, that is the Duffy binding protein, and it also recognized VAR2CSA. So it's a cross-reactive monoclonal antibody. So using this approach, we saw three of these three peptides that were strongly recognized by this monoclonal antibody, but we chose to focus on just one of these peptides known as cross-reactive peptide 1, or simply known as CRP1. Now this figure here is showing the logo plots of this of the amino acid residues in this peptide. So this, essentially what this logo plot is telling you is the degree of conservation of the different residues in this peptide. And you don't see much variation at each position. So this tells you that these amino acid residues in this peptide were strongly conserved. Now this logo plot was generated using isolate sequences from the different parts of the world. So we synthesized this peptide, and the next question we wanted to do was to validate the results of this array. And one way we did this was a simple competition ELISA. So in your direct ELISA, indirect ELISA, where you have the direct recognition by the monocular antibody to VAR2CSA coated on a plate. In a competition system, you pre-incubate the monocular antibody with our CRP1 peptide. And the idea is the CRP1 captures the monocular antibodies. So this leads to a reduction in the signal that could be detected on a plate. So using this approach, we saw that CRP1 was a kind of a major epitope in VAR2CSA that was targeted by this monoclonal antibody. So here is showing the relative binding to VAR2CSA in percentage. These 3 dots represent 3 independent experiments that was conducted. And we see that pre-incubating with the CRP1 leads to a significant reduction in the detection of VAR2CSA compared to our negative control, which is just an unrelated peptide. So this mirrors the result we had from the peptide array, which shows that this CRP1 was a key epitope recognized by the monocular antibody. So the next question we want to answer is if CRP1 was a target of human antibodies from P. virus and P. falciparum exposed population. And what we did that we collected serum samples from non-pregnant population in 2 communities in northern Colombia, known as departments of Cordoba. So we collected these samples, then we screened them for the presence of VAR2CSA antibodies. And of those that were positive, we also screened them for the presence of CRP1 antibodies. So we see that in these 2 communities, that is Puerto Libertador and Terrata, at least 50% of these individuals had antibodies that target CRP1. Now, this is really unique because it means that in this population, they naturally acquire antibodies that target a conserved region in VAR2CSA that is also a key target of a cross-reactive monoclonal antibodies that was generated against the virus content. Now, the unexposed that we see here represent samples we collected from individuals in a region in Colombia where you don't have malaria exposure. So the next thing we wanted to look at is the functionality, because that's really an important correlation of protection. So one way of doing that, we use a binding assay known as inhibition binding assay. Here we have a plate that was spotted with our CSA, which I previously showed is one of the receptors that is known to interact with VAR2CSA. Then we have a parasite that we culture in the lab. So we pre-incubate these infected red blood cells with our CRP1 antibodies. Then we measure the ability of these antibodies to block the binding of the parasite to the plate. But in order to use that strategy, we had to obviously immunize rats with the CRP1. So we generated rat CRP1 antibodies, and we used those antibodies in the binding assay. So here is showing the parasite binding inhibition in percentage. So a higher percentage means that a significant proportion of this parasite were prevented from binding to the plate. So we see when we incubate with CSA, we see about 80 to 90% inhibition. However, when we pre-incubate with the rat CRP1 antibodies, we see about 30% inhibition. So clearly this, it shows that this rat CRP1 antibodies had some blocking effects. So just to conclude, CRP1 was strongly conserved in the feed isolate and was the key target of cross-reactive monoclonal antibody. And we know that in non-pregnant populations in Colombia that are exposed to P. virus and P. falciparum, they naturally acquire antibody that targets CRP1. And CRP1 antibody was shown to have some blocking effects, though in vitro assay. An important implication here is the generation of generate knowledge on the mechanism of cross-species immunity, specifically population that are exposed to P. falciparum or plasmodial virus, and advancement of research novel vaccine candidate that targets malaria and pregnancy, and this time around, targeting conserved regions in protein, which could circumvent the whole issue of protein variants. So just want to use this opportunity to acknowledge members of my lab and also our collaborators in Australia and also in US. And importantly, we have a collaborator in Colombia that are actively engaging with the study participants. And I also acknowledge the study participants themselves, because without them, this study would not have occurred. Thank you.
I apologize. So I apologize, and if I missed this. So CRP1 is a malaria protein.
Yeah.
Have you looked to see how widely it's conserved amongst Plasmodium species or other apicum or other sort of related parasites?
So we looked at so CRP1 is part of this specific domain-like structure known as Duffy-Binding-like regions.
Oh, okay.
Now, those DBL domains, they are present in some P-falciparum parasites, and also some is also present mostly in Plasmodium parasites. And these DBL domains, they can act as evasion ligand, like you have in P. vivas, or a sequestration ligand, like you have in VAR2CSA. So in terms of conservation, the amino residues of this DBL domain, the conservation is low. I would say like 30% is low, but structurally they do look alike. So they contain some similar structural features, like let's say alpha helices or those cysteine domains and stuff like that. Yeah.
Thank you very much.
Yeah.